I teach undergraduates. General biology, I still hear reverberation. I want to get this thing taken care of before we get going here. If this is the culprit, we've got to figure out how to disconnect this guy.
I don't know either.
Nobody knows, right?
Oh, yeah.
We got it's like fused in. Oh, there we go. Okay, let's turn this off. Here, take it. Put it down somewhere. Hide it. All right, let's hope that. Is it better now? Yes. Okay. Good. All right, well, as I was saying, I teach undergraduates general biology that are not majors in biology, and then I teach the cancer biology class for small numbers of students, about 15, and then we discuss the latest papers in the field of cancer. So what I want to do today is review and present some of the information that we've collected over the years, not only from preclinical systems, but also from patients. I'm at Boston College. We do not have a medical school. Cancer is a biological disease, so we study the biology of the disease, and then when we want to make treatments, we have to work with physicians that are in different clinics. So, okay, so I always often start my lectures with an update on the war on cancer that was declared during Nixon's administration in 1971, and just throwing up some numbers from the last few years, 2013 to 2018, just gives you an idea. I used to say the body count continues to rise in cancer. It still does, but what we really have is a crisis for this disease. And you hear papers or people saying that, we're doing really well on the war on cancer. And I think you should look at these numbers carefully because what it says is that we might not be doing as well as people think. So we have the new cases, deaths per year, and deaths per day. And you'll notice that the percentage of increase in simple numbers. Now you can interpret these data in many different ways. The number of people in society, who's getting cancer, older people, whatever. But the issue is the numbers are not going down. That's I think is the. So we have about 1,600 people a day dying from cancer in this country. All right. Now, I came back from China last year, and they have 8,100 people a day dying in China from cancer. That has surpassed heart disease in that country. I've been to Germany and all these different countries, and they're all having the same problem. The number of people dying from cancer continues to increase throughout the Western world. That's the bottom line. So you say to yourself, why is this? Why are we making no major progress? If you have something that works, the numbers you get a precipitous drop, like the AIDS situation. When AIDS was a death sentence, if you remember back in the late 80s, people who got AIDS, the probability of dying within a year was quite high, a year or two. All of a sudden, boom, the drop, because the gay community was a very organized group, and they marched on Washington, and they created a whole bunch of havoc, and the next thing you know, we had the problem managed. Let's put it this way, it was managed. So we have to ask ourselves, we raise money for cancer. The federal government spends $5.6 billion a year on cancer, various types, various research aspects. And then we have all these private foundations, right? Susan Komen and you name them, pink ribbons, this, that, and people are running and jumping and swimming and doing all this stuff for cancer. Let's raise money for cancer. Marathons on TV, stand up to cancer. You hear it, it's everywhere. What do they do with that money? Does anybody ever ask? Where's all my money going? I ran this marathon and I raised all my friends pitched in. How much money goes to cancer research? You have to ask number one. And then the bigger question is, what kind of research are you doing with the money that I gave you? There's no accountability in this. The population in this country has not stepped forward and says, what are you doing with all that money? Because as I said before, the more money we raise for cancer research, the more cancer we get. And those numbers show it. So what's going on? What are we doing? So this is what I want to address, and I want to provide information for why we have these numbers and why things are not likely to change unless we make dramatic changes in the way we look at this disease. So I'm going to give a little cell biology here. This is a cell that we talk about in biology class. The cell, you'll see, has a nucleus and a mitochondrion. I'm not going to get into all the details of all the other organelles, but let's just look at the nucleus, because that's where the genetic material is, the DNA, and that's where all the most of the mutations in the cell are localized in that organelle, the nucleus. And then we have the mitochondrion, that little bean-shaped organelle there. That's the energy generator of the cell. It regulates signaling inside the cell, fluxes inside the cell. So when you look at a cancer cell under the microscope, you see abnormalities in both the nucleus and in the mitochondria, as well as other things inside that cell. So the field today focuses almost entirely on the nucleus and looking at the genetic mutations that exist in the cancer cells inside the nucleus. Okay? That's why they call cancer as a genetic disease, and I'm going to talk about that. But the mitochondria are also damaged in that cell. And energy is everything. Without energy, nothing can live. So you've got to realize that energy is the ultimate determination of life. All right? So cancer cells live, the normal cells of our body live, but they live by different mechanisms, and I'm going to talk about that. So, and as I mentioned in many of my YouTube videos and things, cancer is, the dogmatic view is that it's a genetic disease, and as I said, go to the NCI website, National Cancer Institute, go into their open access website, look down, what is cancer? Cancer is a genetic disease. Well, the federal government can't be wrong, can they? How could that's possible? Our federal government is wrong? That's saying to everybody, well, maybe, you're going to find out. So the hallmarks of cancer cells carry the oncogenic and tumor suppressor genes, mutations that define cancer as a genetic disease. And as I'll show you from textbooks, it's everywhere. Textbooks, the NCI. A dogma is an irrefutable truth. It's like a religion. You don't question your faith. So this is what we have in the cancer industry. The cancer field, not just the industry, the field of cancer biology and whatever, it's a genetic disease. Thousands and thousands of scientists in our country and around the world are studying genetic mutation differences in cancer cells.
If you want to learn how to implement the ketogenic diet and the Press Pulse protocol into your cancer management, just visit school.com/keto for cancer, the new community by our research analyst Johnny Rockermeyer. Within this new community platform, you can connect with other patients and learn from their experiences. You can also just search through the posts other members have already made. You will have access to our classroom section as well, which provides practical advice and tools for the best results. These sections cover all parts of the Press Pulse protocol that you should implement. Starting with the General Scientific Concepts by Dr. Thomas Seyfried, we will show you how to start the ketogenic diet, including which foods to choose and which to avoid. Additionally, we prepared a list of doctors specialized in metabolic therapy, as well as details about glutamine inhibition and hyperbaric oxygen protocols. So, take a look around at school.com/keto for cancer.
Now here's a picture from a textbook that we give in general biology to try to educate people into letting them know. It's called the somatic mutation theory. That's the theory. In science, a theory is not a guess. Okay, when somebody says, I have a theory about something, that's like a hunch. No, in science, a theory is supported by massive amounts of evidence. Theory of relativity, Einstein, right? So this is supported by Darwin's theory. You have theories that are supported by massive amounts of information. The somatic mutation theory is the structure upon which this disease is viewed. So you have this car, he's speeding out of control and he has no brakes. And the out of control speed is due to mutations in proto-oncogenes. And the no brakes means there's mutations in tumor suppressor genes. And the bottom line, cell division out of control. So people say, what is cancer? Cancer is cell division out of control. That's the bottom line. I have a group of cells and they're dividing out of control. Why or how are they, what's making them out of control? Mutations in oncogenes and tumor suppressor genes. Okay? And this is what reflects this. Now, when we talk about somatic mutations, how do we get cancer? So we have a mutation in a cell. Here's a normal cell on the left and a malignant cell on the right. So we got one mutation in this chromosome, another mutation in that chromosome, and in this and that, ba-bam, ba-bing, ba-bang. And next thing you know, we take this normally controlled cell and we make him growing out of control from accumulation of different mutations, somatic mutation theory. No one knows for sure how many mutations, 1, 2. Some cancer cells have thousands, millions of mutations. Other cells have no mutations. There's some cancer cells have no mutations. Nobody talks about those. Then here's the quote from Dr. Vogelstein at Johns Hopkins. We know now precisely what causes cancer, a sequential series of alterations in well-defined driver genes, okay? We also recently, within the last couple of years, interestingly enough, scientists from Britain and Japan have found a number of driver genes in normal cells in the esophagus and skin. So how can the disease be caused by driver genes that exist naturally in your normal cells that are not dividing out of control? This is a shock wave now in the field. Nobody, whoa, what are you talking about? Driver genes in normal cells? There's some tumor cells that have no mutations? So this is really an issue. So the question now becomes, who started this whole concept that cancer is a genetic disease? Where did it come from? It came from this gentleman here, Theodore Boveri, speculative essay on the origin of malignant tumors, 1914. 1914. This young scientist was looking at sea urchins and noticed there were some chromosomal abnormalities in the sea urchins. And he said, whoa, this could be the origin of cancer. And here he says, I have no personal experience worth mentioning any of the numerous specialized fields of tumor research. My knowledge comes almost exclusively from books. Given this, it is inevitable that I am unaware of many reports in the literature that I overestimate the significance of many known facts, and I do not set enough store on others. But this article will doubtless contain even more serious, what does it say, serious defects, as is so often the case when an author makes an incursion into a field in which he is unfamiliar. Can you believe it? Now, where do we go? Today, personalized medicine, personalized therapy, precision medicine, all based on the gene theory of cancer from Boveri in 1914. Right? This woman is looking into a screen. They took a biopsy from a breast tumor and looking at the gene mutations on the screen here to try to figure out whether they're going to make a prediction on prognosis or treatment based on a spectrum of gene mutations, which I'll show you are almost completely irrelevant to the nature of the disease. These screens cost $7,200. The company is here in Cambridge and Boston, where I am. Right? They just bought it out, $4.4 billion for a company that screens mutations that are largely irrelevant. Makes people, oh, I got this mutation, got that, can you help me? Forget it. I'm going to show you evidence. And I have here observer effect. What does that mean? And this is what I spoke about last night about the biopsy material. There's a lot of papers published in the literature. Okay, you have a benign tumor. Why stick the benign tumor to tell the person you got a benign tumor? If the tumor is malignant, why stick the malignant tumor? Because you could possibly set it loose. There's a risk for this. It happens in brain. I'll show you. The brain is brutal. It happens in liver cancer, potentially breast, colon cancer. A lot of these cancers, when you start screwing around with them, you change the microenvironment, and bang, you send out these cells into the lymph nodes and bloodstream. Why do that? So you can get a gene screen. And I'll show you. So what? It's irrelevant. Yet they do it. They do it all over the place. So let's talk about evidence that challenges this whole idea that cancer is a genetic disease. And whenever you challenge dogma, you're going to be up for you don't get attacked, I would say. Most people ignore it. This doesn't mean anything. Obviously, Dana-Farber can't be wrong. MD Anderson can't be wrong. Sloan Kettering, and most of all, the federal government can't be wrong. So people do this. They have these kinds of responses. But when you go back and look at the scientific literature like I did, You look at these studies that they have done, like this is a frog with a giant kidney tumor. The arrow is pointing to this giant kidney, a very aggressive tumor, kills the frogs. Dr. McKinnell and his colleagues in 1969 published in Science, the top leading journal in the field, isolated the individual cells from this renal tumor, took the nucleus, that little circle, and put it into an enucleated egg, and cloned let the egg develop into a tadpole. That tadpole, figure 2 there, shows a tadpole cloned from the nucleus of this very aggressive kidney tumor. Now, what happened to all these driver genes? What happened to the dysregulated growth? It wasn't there. And he looked at every one of the organs, cut the tails off, they regrew all of this from the nucleus of a tumor cell that's supposed to have driver genes. He concluded that the findings are inconsistent with the somatic mutation theory. Okay, this is a body blow to a dogmatic view of what the field is. Not consistent. This is work done by Dr. Rudy Yanish at MIT, and he showed that you could clone a mouse embryo from the nucleus of a melanoma, and he characterized the mutations in the melanoma nucleus before he cloned the mouse from that nucleus, and he found the same mutations in the mouse that had no dysregulated cell growth. How do you explain that? He says the presence of the genetic abnormalities in the embryo provides unequivocal genomic evidence that the mice were in fact cloned from the tumor nucleus, but you don't see the dysregulated cell growth, the singular phenotypic aspect of the disease. How do you explain that? On the somatic, well, we ignore it. We don't talk about it. This is work done by Li Jun Wang at Baylor College of Medicine and her colleagues. And what they did was they transplanted mitochondria from one cell to the other. So you take a very aggressive human breast cancer cell, growing like crazy. You remove the mitochondria, and you put the mitochondria in from a normal cell, and that very aggressive breast cancer cell then becomes growth managed. Now, if you take the mitochondria out of the very aggressive cell and put it over into the indolent cell, that indolent cell now becomes very aggressive, telling us that it's the mitochondria in the cytoplasm that seems to be playing a much bigger role in the determination of the growth than does the nucleus. And I wrote this paper, and all I did was pluck these findings out of the literature and put them into a single paper. So now it's hard to ignore them. When you put them all together, hundreds of articles, hundreds of experiments placed into a single document, comparing and contrasting the results on two competitive theories. One is the mitochondrial metabolic theory, and the other is the somatic mutation theory. And then I let every person with a rational mind read. It's open access. Anyone can go to the PubMed, go to the Google, you can get these papers. And you can read for yourselves and let you make the decision. Don't listen to me. Read the data, look at the experiments. Sometimes they're a little complicated, but a lot of them can be easily understood by the lay public. They can read these things too and say, wow, how do you explain that when you do it for yourself. So, and this was the final summary of all this. So you have the green cell and the red cell. The green cells are normal, and they can be so you have normal nucleus, the round nucleus, normal mitochondria, the little bean shape with the cristae, the little stripes. The red cell produces more cancer cells, and they have mutations in the nucleus. We're not arguing that they do have, many cancer cells have loaded with mutations. But they also have damaged those little red beams. They don't work right. And more cancer cells get more cancer cells. But then when you do the experiment, take the red nucleus and put it into the green cytoplasm, you get normal cells, despite the fact that the cancer nucleus that supposedly has the driver genes is still there. You saw the frog, you saw the mouse. I don't have time to show you all the other organisms that were changed like this. And then you take that green nucleus and put it into the red cytoplasm. Now this is the normal nucleus, goes into the red cytoplasm, and you get dead cells or tumor cells. You don't get normal cells. Man, it's telling us that it's the mitochondria in the cytoplasm that's the driver of this disease and not the nucleus. The whole field is on how many gene mutations do you have? Irrelevant. No, so if somatic mutations are not the order, how do we get cancer cells? Okay, this is the guy right here, Otto Warburg, 1920s and 30s, Nobel Prize winner. he defined cancer as rises from damage to cellular respiration, which is inside those mitochondria. So he said, this is the origin of the disease. Everything else is secondary, he said. Everything else is secondary. Mutations, virus, anything, everything else is secondary. It's the respiration of the cell that's the origin of the disease. So, if respiration is defective, as I said, everything is energy. How do you get a cell to live if I take away its respiratory energy? And therefore they start to ferment. Fermentation is an ancient metabolic pathway that existed on the planet before oxygen came into the atmosphere. So these cancer cells are fermenters. They ferment to get energy. They don't respire because the organelle, and I'll show you the evidence, is defective. Cancers continue to ferment lactic acid in the presence of oxygen, which is weird. You shouldn't be fermenting when oxygen is present. You ferment when there's no oxygen, but when oxygen comes into the environment, you should stop fermenting. Cancer cells continue to ferment even in the presence of the oxygen. So that tells us that there's something wrong with their respiration. The red I have because we just published a paper. You have no idea, because unless you're really engaged in the field, the controversy that Warburg stirred up by his... So they threw him under the bus, as I said last night, because Watson and Crick discovered mutations in the DNA, and that was sexy, and everybody thought that was the origin of the disease, and we have the crisis in cancer that we have today as the result of that huge intellectual shift. But Warburg's theory was not completely credible to some people, because they said, if Warburg is right, how is it possible that some cancer cells can grow without glucose? Therefore, Warburg is wrong. And how come some cancer cells take up oxygen? That means Warburg was wrong. And everybody said yes, because God forbid Warburg was right, so we have to say that Warburg was wrong. We just published a paper, it just came out in ASN Neuro, the red. We also found for the first time, and we identified for the first time, that these cancer cells also ferment an amino acid called glutamine. So this now is the missing link in Warburg's central theory. These cells can ferment not only sugar, but they can also ferment sugar, glucose, and glutamine. And Q is this designation for glutamine, and it's the missing link that we have now found, and that showed that Warburg is right. Warburg's theory is correct. He didn't have all of the parts of the puzzle, but he was fundamentally correct in defining cancer as a mitochondrial type of problem. So, and listen to this. If you take cancer cells and do the gene screens, every cell in that tumor has all kinds of different mutations. But every cell in that tumor, regardless of what the tumor is, they're all fermenting. Fermentation is the common metabolic problem in cancer. It's a fermentation metabolism, and it exists in all types of cancer cells, so that means all cancers are essentially the same disease. Some people have a cancer in their brain, some people have a cancer in their colon, in the breast, in the bladder, in the lung. Whatever it is, it's still fermenting. It just happens to be a different cell that's fermenting. The mechanism is the same, they're fermenting. Warburg was right. We have established that Warburg was right. And here's our paper. I don't have time to go through the mechanics of this. It's quite detailed into the biochemistry, but you just like to show people that you did in fact publish a paper showing that mitochondrial substrate level phosphorylation, which is the missing link in Warburg's theory, ultimately underlies not only glioblastoma, but also other cancers as well. And we have many of those short diagrams. I'm not going to go into that. But this is easy. Everybody can see this. Normal mitochondria has those nice stripes. Those are called cristae. Those stripes contain the proteins and lipids of the electron transport chain, which allow us to get energy when we breathe. Those stripes mean we're alive. Our cells are breathing. Now, if you look at the glioblastoma mitochondria, very aggressive brain cancer that I'll talk about, you see the stripes are missing. The cell can't, the organelle can't get energy from respiration because the very structure that's needed to do that is defective. Now here I'm just showing you more. Like, you have to see this under the electron microscope, because a lot of people don't look at these under the when you look at cancer cells under the electron microscope, you see all kinds of abnormalities in the organelle. So here on the this is a breast cancer. On the far right there, you see the stripes. You can see them in that kind of cell. And the one on the left, you can see them broken around, holes and things like that. And on the bottom, that's colorectal cancer. And those little arrows are pointing to mitochondria that have no cristae. Again, in biology, we have a very powerful tenet. Structure determines function. You've all heard it. Structure, function, structure, function. If the structure of the organelle is defective, the function of the organelle is defective. It's so simple. Now, for people to say mitochondria are normal in cancer, I went through all these different cancers and looked at all these under different electrons, and they're all defective. in one way or another. Pete Peterson showed this years ago. There's another very simple diagram, energy metabolism in normal cells. All you have to do is look at the big yellow starburst there, ATP. ATP, adenosine triphosphate, is the energy currency of the cell. CO2, and so when we breathe air, taking a deep breath, we're all alive here, right? You blow out CO2, okay? CO2 is a waste product, normal respiration. Everybody's respiring here, we're all breathing. Water is also a waste product. We build urea, and we build water, and we eliminate the wastes. And CO2 is a waste product from the foods that we eat. And we produce very little energy from substrate level phosphorylations in the cytoplasm and in the so-called Krebs cycle. This is the way most of us are alive and doing this. Now you look at a cancer cell, the same diagram, but you see the big starburst is now coming out of a different location. It's coming out of that Krebs cycle in the middle of it. It's not using electron transport because I just showed you the evidence that the structure of the organelle that is responsible for the electron transport is not there. So the cell still gets energy, but it's coming from ancient pathways. These pathways existed on the planet in organisms before oxygen came into the atmosphere. These cells are doing nothing but falling back on ancient pathways. And once they fall back on the ancient pathway, they become dysregulated in their growth. Okay? Growth dysregulation comes when you shift from respiration to fermentation. And you can see the distributions of this. So people say, how do you get cancer? People say, how do I get cancer? How do you get cancer? So we have carcinogens, radiation. We talked about that. You get exposed to a carcinogen, you're going to get cancer. These are, well, I call these risk factors. People can work in a very toxic environment. 50% of the people will come down with some sort of a cancer, but 50% don't. So it's a risk factor. It determined from your genome determines whether that risk factor will elicit a cancer. But the genes mostly protect us. Radiation, hypoxia, that means this absence of oxygen in a population of cells can damage the respiration, cause cancer. Inflammation, systemic chronic inflammation is a risk factor for cancer. Rare inherited mutations, like the BRCA1, you hear about Angelina Jolie and these people having their breasts and ovaries removed because they have the gene. Only 50% of the people that have that gene actually go on to get the cancer. It's a risk factor, it's not a determinant. The gene will cause the cancer only if it damages the mitochondria. If the gene does not damage the mitochondria, that person will not get cancer. RAS oncogene viruses, hepatitis C, papillomaviruses. These viruses cause cancer because the virus goes into the mitochondria, damages the respiration, and you get the cancer that way. So this is what's called the oncogenic paradox, and it plagued the field right up until today. People say, I don't know what the common pathophysiological mechanism is that we get cancer through a common mechanism. So we have a lot of risk factors that can produce cancer, but they all come through a common pathophysiological mechanism, which is damage to the respiration. Those ROS, R-O-S, are reactive oxygen species that are known to be carcinogenic and mutagenic. What does that mean? ROS cause mutations, ROS are carcinogenic. They damage the respiration further, and they lead to the gene mutations that people study in the nucleus. So the mutations that people are studying in the nucleus of the cell are secondary downstream effects of the damage to the respiration. They're not the cause, they're an effect. Now think about that. Think about that when you hear all these talks on the TV about this drug and that drug and all this kind of stuff, because they're all based on the gene theory. Opdivo, Keytruda, all those things, these immunotherapies are based on the gene theory. Think about that. Now in his book, Emperor of All Maladies, Sid Mukherjee, whose book was on the New York Times bestseller list for about a year or more, Pulitzer Prize book, Emperor of All Maladies, he struggles with the oncogenic paradox. Doesn't mention Warburg at all. He struggles just like everybody else. All right? If they understand the biology of the disease, they will understand what the problem is. So, the ROS then damaged the respiration. Now, the diagram on the bottom, you see the green line going down and the red line going up. Okay, the green line is the breathing capability of the cell. The red line is the fermentation capability of the cell. So cancer is nothing more than a transition away from respiration to fermentation, which can be caused by any number of provocative factors, either in the genome or in the environment. They're all risk factors. They're all what they call secondary causes. The primary cause of cancer is damage to the respiration within the cell, leading to genomic instability, and the oncogenes that people love to talk about are transcription factors that open the floodgates to bring in fermentable fuels into that cell, because if you don't, respiration is a remarkably efficient way to get energy. Fermentation is a massively inefficient way to get energy. So if I'm going to drive the fermentation machine, I have to bring in large quantities of fermentable fuels that the cells can ferment. And those fuels are glucose and glutamine. The two fuels, Warburg always knew about the glucose. He didn't know about the glutamine because the structures that were needed and the pathways were identified only after his death. So he didn't know the missing link. We now know. So on the right hand, you see all these hallmarks of cancer. Every one of those hallmarks is the subject of tens of thousands of investigators involving tens of millions of dollars investigating all these hallmarks of cancer. I'm not going to go through them all, but the one that's why cancer cells evade programmed cell death. If we stab ourselves, we get a lot of dead cells. The cells die naturally. Okay? They don't have oxygen and they die quick. Cancer cells don't die. Why is that? Because the organelle that controls their life and the life of our cells is the mitochondria. It is the kill switch. When the mitochondria become abnormal, the cell dies through a very interesting biological series of events. But if the kill switch is broken, the cell continues to grow out of control. So all of these hallmarks. Now, the big dog here is metastasis on the bottom, number 6. And that we have to work into the whole concept of energy metabolism, because metastasis is ultimately the death, what kills most people. As a matter of fact, like I said last night, it's not clear whether it's the metastatic cancer that's killing the people or whether it's the toxic treatments that are being used to try to manage the metastasis. We don't know that. Okay, so we can't be sure, but it's metastatic. Once your cancer starts spreading around, you get nodules all over the place, that's called metastatic cancer, and that's ultimately. So where does that come from? Okay? And here I'm showing this nice little diagram. You see the cells, the blue cells on the left are all standing in a row. They're all nice and neat. They're all columnar. They're very attached to each other in the basement membrane. Any one of those provocative effects from the environment can cause a destabilization in the growth of these cells, and they start to disturb, they start to grow. The red cells that you see there, that's our immune system. Our immune system surveils our body all the time. Those little cells now are growing a little bit dysregulated, and they cause a disturbance in the microenvironment. It's like having a house fire in the neighborhood. Ooh, what's the smell? Smoke? Well, something's going on. Call the fire department. Fire department comes and hoses down with water, hoses down the house. Well, these red cells do the same thing, but it's out of context. They throw growth factors and cytokines to try to stop the environment, to try to stop this inflamed, growing environment. And those factors are, it's like the fire department throwing gasoline on your house. The fire gets even worse and burns down the neighborhood. And that's what's happening here. These cells aren't smart. They're genetically programmed over millions of years of evolution to perform specific functions. And if that function is done out of context, you get into a big problem. So as you can see, these cells are very fusogenic. So what they do to try to put out the fire, they fuse with each other and with the other cells. Now what happens over time, the red cells gain abnormal mitochondria from the cytoplasm of the cells that are dysregulated. Now those cells, the red cells, already have the capability of moving in and out of our body. If I cut myself here, I've got local immune cells to try to fix, and I got monocytes in my bloodstream from the spleen and thyroid gland, and they come flying into this spot to try to heal the wound. So they move in and out of our system all the time. So these red cells are now gaining the abnormal mitochondria from the cancer cells, the tumor cells. The tumor cells themselves can't spread. They don't have the capability of moving in. They're localized in that spot. But once those red cells now become contaminated with the abnormal mitochondria, they are already genetically programmed to spread through your body. Okay? And they suck down glutamine like there's no tomorrow. They're cells of our immune system. And I said last night, the immune system uses glutamine. Our normal red cells, and now this corrupted tumor cell, which is part of our immune system. The metastatic cancer cell is us. It's our powerful immune system gone rogue. They live in hypoxic environments. That's why the angiogenic field doesn't work. Avastin and all those crazy things that people talk about. You can predict. When you understand the biology of the disease, you can predict all the stuff that's going to happen. And then they spread and they set up. And I don't want to get into the EMT and all this other stuff that my colleagues in the academic community talk about. But this is the origin of metastasis. So if most cells obtained energy through ferment, what are we going to do to stop the tumor? We got to take away their fermentable fuels. One strategy is you target the fermentable fuels and you elevate non-fermentable fuels. And here's you got calorie restriction and therapeutic ketosis that I talked about last night. So this reduces glucose tremendously. It maintains minerals and nutrients. TK, therapeutic ketosis, reduces glucose. If tumor cells, to burn ketones for energy, you got to have a good mitochondria. I just showed you evidence the mitochondria are dysfunctional in cancer. And also, ketone bodies, for those people who don't have cancer, if you get into therapeutic ketosis, your mitochondria get super healthy, and the likelihood of getting cancer goes way down. All right? You can't get cancer unless your mitochondria are damaged. So how do you prevent cancer? Keep your mitochondria healthy. How do you do that? Do therapeutic fasting, calorie restriction, this kind of stuff. Not easy. And water-only therapeutic fasting, right? Oof. All right. Okay, that's enough. I don't want to talk about that anymore, right? Then these ketogenic diets, such a misunderstood field. It's not, it's therapeutic ketosis. But how do you get into therapeutic ketosis? Well, one way, you can stop eating for 3 weeks, 7 days, and then stay on it. Oh, it's not good. It works wonderfully, but it's not very hard. Ketogenic diet's a very massive misunderstanding in the field. It's not an Atkins diet, and you eat little bits of it. You don't gobble down a whole bunch of butter and lard and that kind of stuff. Anyway, very low carb, very high fat. It must be done in restricted amounts. I worked in the epilepsy field for years. Every now and then, one out of 200 or 300 kids loves to take the oil, puts it on everything, and his seizures don't go away. Ketogenic diets are remarkably effective at managing seizures in children, but you have to be rigid about it, and you have to keep the blood sugars very constant, very constant. And if you add too much fat, you get insulin insensitivity, and the sugars go up. So people have to know how to use this. It's a tool. This is medicine. We're not talking like we use drugs. A physician applies drugs to your body because he doesn't want to give you too much, it'll kill you, and not enough won't do anything. This metabolic therapy is the same, and every person is a different entity, so you really have to know what you're doing before you go out and do this stuff. And you've got to have knowledgeable professionals that will help you. Here's the simple, bring your blood sugars down and bring your ketones up. Ketones come from the metabolism of fats in our bodies, and then you can get into the state of managed growth. And I'm not telling you we can cure cancer by doing this. I say we shut down their ability to grow as fast as they would normally grow. It's a big difference than saying you can cure cancer with a keto. You can't. I don't want to say you can't, because some people are doing very well, which I met from reasons I don't know, but I would be shocked. So to help patients, at my lab, we developed the glucose ketone index calculator, published in open access journal, Nutrition and Metabolism. Therapeutic efficacy is considered best with an index value of approaching 1.0 or below. You get a little meter from Amazon, glucose ketone meter. There's a couple of them, Precision Extra, Mojo, Keto Mojo, and these things. And you can, at home, you can take the meter, take the drop of blood, put it on the glucose stick, just like someone with diabetes. Oh, my blood sugar is there. Pull that stick out, squeeze your finger again, put the ketone stick in, put that, I got the ketones. Now I have to just divide the ketones by 18 to get millimolar, and that's the calculator. And the next thing you know, you get your ratio. What's your ratio? People say, can I eat this? Can I eat that? I don't know. What's your ratio when you eat it? Oh, I didn't know about the ratio. You got to know the ratio, because the ratio tells you whether you can eat that or drink this or whatever. Once you know you're in therapeutic ketosis, then you build your diets around your ratio. You understand what I'm saying? Okay, now, this was one of the first studies that was done in children with two little kids, hopeless cases, massive brain damage from radiation and chemo. They were both given up. And Linda Nebling at Case Western took these kids, and the physicians at the time said, yeah, go ahead, try this. And both kids responded remarkably to reduced blood sugar, elevated ketones. One child lived far longer than was expected, and the other child was lost to follow-up. So when that means that they were wired, and then she just moved somewhere else and nobody knows what happened. But this is the first and only time we've adapted this to children pediatric oncology. It's never been done before. Yet we're doing the same thing in the epilepsy clinics all the time. Little kids with seizures, we use these metabolic therapies to manage their seizures. But God forbid we take a kid that has a brain tumor and do the same thing. Linda did it, opened our eyes, so we followed up and we did it in the mice. So this is a preclinical study on a glioblastoma cell in the mouse. SDUR means standard high carbohydrate diet. Let the mouse eat as much as he wants. Eat as much as you want. Very high carb diet. The standard diet restricted. Just restrict the diet to reduce 40% of the calories. And you get therapeutic ketosis from that. And we got a 60 to 85% reduction in the size of the tumor. You can see the guy on the right, his head is less swollen than the guy on the left, just by restricting the calories. What went on here? Then these little black squares, each mouse is a square is a mouse under a different dietary condition, right? So and you look down on the bottom, this is a linear regression analysis, and if you look down on the bottom, as glucose goes down, beta-hydroxybutyrate, BOHB, is a ketone body. That's what happens. When you stop eating, the brain needs energy. We start mobilizing the fat out of our body stores, and they get converted into beta-hydroxybutyrate, a ketone body, and replaces glucose for our brains. Evolutionarily conserved adaptation. Human beings evolved to starve. If it weren't for our ability to live long periods of time without food, none of us in this room, myself included, would exist today. The mice can't do what we can do. Mouse will die in 5 days from starvation. Humans can go very, very long before they die from starvation, depending on how much body fat you have. Now on the right, you see tumor weight and the glucose. So as glucose levels go down, the size of the tumor goes down. The higher the blood sugar, the faster the tumor in these mice go. The lower the blood sugar, the slower the tumor grows. Very clear. This has now been replicated for humans with brain cancer and colon cancer and breast cancer. If you want your cancer to grow fast, do everything you can to make sure your glucose is as high as you possibly can get it. How do we do that? We eat cake, and we eat ice cream, and we eat Infamil, and we eat all these different kinds of things that they give to the cancer patients in the clinic. Right? You go down to the oncology, eat whatever you want. Don't you know that there's a large number of papers in the literature that say that your cancer will grow faster if your blood sugar is higher and will grow slower if your blood sugar is lower? No, I never heard of that. These are the data. And I've referenced from a vast amount of human data. It was based on our work in the preclinical study, and then people looked at it in humans and they found the same thing. Also, when you get your blood sugars down and ketones up, it becomes anti-angiogenic, which means the blood vessels go down inside the tumor, the angry blood vessels, the tissue becomes less angry, less inflamed, and becomes pro-apoptotic. You start killing these cancer cells. There's no drug in the pharmaceutical armamentarium that will do what this therapy will do. But it requires personal discipline. It's not easy to do this. It's much easier to sit there and get infused with some toxin or get your head irradiated or body irradiated. But these are the evidence. It's published. Now I'm going to talk about glioblastoma. This is a terribly aggressive tumor, and we all know it's poor prognosis, no effective therapies, multiple cell types. This is the tumor that just recently killed Senator McCain, before Ted Kennedy and many others. Highly invasive. It spreads outside the... So let's talk about this, because this is one of our major focuses in the lab. If you manage this guy, you can manage anything. This is a cross-section through someone's brain who perished from GBM, glioblastoma. You see the big cystic spot. And if you notice, there's a midline in the brain from the top. You see that line, and it gets curved out to the left. That's the pressure from the growing tumor pressuring the ventricles. Most of these poor folks die from intracranial pressure. You can see the big ugly cyst. Now, the problem, you can't cut these guys out because tumor cells use the blood vessels as a kind of a railway system to get through the brain, and they go across the top of the blood vessels at one of their ways of invading. And those little purple cells on the right are cancer cells on the top of blood vessels, and what it is they can just, so you don't see them on these big scans. You only see this necrotic large area when you do a scan. You can't see these tumor cells that are all through your brain, and this is one of the reasons why it becomes very difficult to manage this disease. Now here's mitochondria are abnormal in glioblastoma. So here, normal, you see on the left, the lighter color bars are the normal, and the GBM have much fewer of these normal-looking mitochondria. And then abnormal mitochondria are enriched in the GBM cells. This is exactly what I said before. The cancer cells have damaged mitochondria. What does that mean? It tells us right away, those cells are fermenting. If you can't respire, you must ferment. There's no other way you can get energy if your respiratory system doesn't work. Okay? So the cells ferment. That tells us, that's the clue. The cancer cells are fermenting. And what they ferment glucose and glutamine. So those are the two fuels. And you know what's very interesting? When the tumor cells suck down a lot of glucose and glutamine, they become remarkably resistant to radiation and chemo. Okay, they say, well, the cancer came back. Oh, it was resistant. It was resistant because they're fermenting. What happens if you take away their fermentable fuels? Oh, you can kill them. So this, I'm not going to go through the mechanics. They get energy from fermentation metabolism using two primary fuels, glucose and glutamine. They can't grow without energy, period. They can't use ketone bodies because their mitochondria are defective. Transition the body over to ketone bodies. Your normal cells can use them, but the cancer cells can't. So that means we've marginalized the enemy. We've put them into a box. And now we slowly degrade them by targeting glucose and glutamine. Now, what do we do in the clinic when somebody is diagnosed with a glioblastoma? Okay? As we heard from Pam and we heard others last night, shock. The first thing that happens is total shock. Not only to the patient, but to the patient's family and friends. Because immediately they rush to the web and they say, what is this glioblastoma? Oh my God. You look at it and you go, oh Jesus. So before you know what to do, they got you in the operating room, and sometimes that's necessary, sometimes it's not. And they've got you scheduled for radiation, and they've got you scheduled for chemo. And by the time you wake up, you're already, and I'll show you what's going to happen. We did the neurochemistry. We defined the neurochemistry. Now, people say you need clinical trials to prove this. The biochemistry tells us what's going to happen. The neurochemistry of the brain, we can make a prediction as to exactly what's going to happen based on what we know, the energy metabolism of normal cells and tumor cells. So here you have this brain, and the tumor is cut out, and when you cut that tumor out, you leave a beautiful hole, a very inflamed hole, and then you irradiate, you break apart the very tightly linked neurons and glial cells. They're all in this beautiful society, very interactive, and now they spew out their glutamine into the microenvironment, and those tumor cells suck it down like there's no tomorrow. You created a Garden of Eden of fuels for these surviving tumor cells that were not first moved out from the surgical resection. Now, when your head is irradiated, your brain tends to swell, and the area tends to swell. So what they do is they give you high-dose dexamethasone, which is up on the top there. Dexamethasone induces hyperglycemia. That means high blood sugar. Now, so we get a massive amount of glutamine being released by the surgery and by the radiation, and we get a higher blood sugar. I don't have the time to tell you, there's a whole bunch of other things that make that environment for the tumor the best growing environment. It's like taking grass and putting flowers and putting every kind of a fertilizer on it. It explodes and grows, and it's vibrant. So we can predict, without any degree of certainty, that this therapy will lead to the rapid recurrence and the death of the majority of people that receive this therapy. I don't have to say any more. I just said, well, where's the evidence? Right here. Now this Stoop, Dr. Stoop, defined the standard of care for glioblastoma. Radiation, chemo, and radiation temozolomide, and now he's coming out and saying maybe steroids are no good. But if you look at the red line are those poor souls that were irradiated alone. Right? They're all dead. This is data. These are the data. I could predict this based on knowing the biology of the tumor. You don't need a clinical trial. You just have to understand the biology and neurochemistry of what's going on. You can predict what's going to happen. And then here's the evidence to prove that you knew what you were talking about. These guys are all dead. Not a single survivor in this study. Predictable. They're dying from the radiation treatment.
Are you passionate about the groundbreaking and heroic research of Dr. Thomas Seyfried on metabolic therapy and cancer? So are we. That's why we've created something special for you, in collaboration with Johnny Rockermeyer, a German book publisher and translator. Introducing our collection of meticulously crafted books that distill the essence of Dr. Seyfried's work. Dive into the science and discoveries. These summary books are your gateway to understanding the intricate world of metabolic ketogenic therapy in a clear, concise, and engaging way. Whether you're new to the subject or a seasoned enthusiast, our books offer insights that can change your life. Ready to explore this transformative knowledge? Visit our website at www.cancerasametabolicdisease.com to get your copy. You can buy the ebook there directly and the paperbook via the provided links. Here's the best part. A portion of every purchase goes directly to support Dr. Thomas Seyfried's groundbreaking research. That's why the direct ebook purchase is the best option to donate as much as possible. You can see all of the donations Mr. Rockermeyer has already made at www.ketoforcancer.net. That's right, when you buy our books, you're not just investing in your own knowledge, you're also contributing to the future of cancer research. Help us make a difference. Together, we can drive change and save lives.
I'll tell you more about that. Now, the little blue line was the biggest breakthrough in glioblastoma in 75 years. What did they do? Temozolomide. It's a toxic alkylating agent that increases driver mutations, which is even more absurd. Okay, temozolomide, nausea, vomiting, fatigue, and diarrhea. Indirect forms of calorie restriction. No one's done a controlled experiment to test what I just told you. Who's going to do that? Here's an example of, let's put faces on the tragedy. Brittany, diagnosed low-grade tumor in January 2014. Low grade. Doctor's very optimistic. Oh, you have a low-grade tumor. 3 weeks, boom, glioblastoma. This is what I'm talking about. You touch the beast will bite you if you don't know what you're doing. We have what they call secondary glioblastoma. These are people who go on and get glioblastoma from trying to cut out a low-grade tumor. You create the environment, this tumor morphs into the big daddy. So then she goes on standard of care, and you can see her on the right. She's got the moon face. She doesn't look anything like the one on the left. That's from overdosing steroids. High-dose steroids cause moon face, thin skin. So she looks at the data, and she says, I'm out of here. I'm going to die. I'm going to Oregon. She's from Northern California. She goes across the border, and she dies with dignity with her family and her new husband. And she writes it all up in People magazine, so you can go back and read People magazine and get the full... Nobody's talking about what put her in that state in the first place. It was, is it moral to take your own life when you have a terminal disease? That was the big subject of the story, not the fact that radiating the brain put her in that position in the first place. Here's Pablo Kelly. He hit me. He had a diagnosed GBM in 2014. He's an English guy from Devon, England. He emailed me and he said, you know, they told me I have a GBM. They want to immediately do surgery, radiation, and chemo, and I don't want to do it. So I said to him, well, you might want to try to get yourself into therapeutic ketosis. So he says, okay, goes into therapeutic ketosis. I hadn't heard from him for 2 years. Didn't know what happened to Pablo. He emails me in 2016, tells me he's doing well. He's thinking about maybe getting his tumor resected because he had an inoperable, now it all of a sudden became operable on 2 years of therapeutic ketosis. No surgery, no radiation, no chemo, just metabolic therapy. I saw him just in September when I went to England to give a lecture to the Childhood Cancer Center. He's doing fine. His wife just had a baby. You know, he's trying to raise money to get fat foods and things just to keep himself alive. And they ignore Pablo. Well, he shouldn't exist. No, Pablo should exist. And many more Pablos, and I know very many more, will be just like him if you avoid the thing that's going to put you in the grave. Right? It's biochemistry, it's neurochemistry. You got to know what you're doing. I don't know if Pablo's cured, but he's still alive and he has an enjoyable quality of life. I mean, he's in ketosis, which, you know, he's not eating pizzas and donuts and stuff like this. But he certainly measures his blood sugar and he does just what I told you. Is he one? No, we have many more like this. Now, we did a lot of work on preclinical studies. I want to share with you just briefly the studies that we're doing here. How do we get to knowing about all this? Because we do everything preclinically. I would like to, before I would be tested on anything, I would like to have someone, did that mouse, or did he do okay? No, he died quick. Well, I don't want that. You know, you take a dog and a cat. How's the cat doing? The cat's doing fine. Oh, okay, that's pretty good. So, yeah, maybe you should think about that. So, and the funny thing, you hear these people say, well, that stuff is done in the mouse. Yeah, you damn well it's done in the mouse. You don't want to be experimenting on humans before you do it. Yeah, so we do everything preclinically. We built the best animal models for these diseases. So we can predict what's going to happen when we go to the clinic, just like we predict what's going to happen when you irradiate your brain when you have a GBM. So and this is what brought us onto the glutamine issue. Because at first I was on Warburg's camp completely. I didn't know what was going on in the other half until we started to do this stuff. So on the left, these dark purple cells are ripping through the brain, invading right across from one side to the other. Then you see the one on the right. This is the hippocampus part of the brain. Calorie restriction or ketogenic diets pounded this tumor, pounded. And we stopped the invasion, but we couldn't kill all the tumor cells. And man, we did everything to hit those glucose. We just couldn't stop it. And then I said, what's the other fuel driving the beast? It's glutamine. We're not targeting glutamine with this metabolic approach that we have here. So you got to target both fuels simultaneously. So we use this drug just to test concept, proof of principle. There was a drug that's known, 6-diazo-5-oxo-L-norleucine, DON. It targets glutaminase. It's an enzyme that breaks down glutamine so that a cell can use the energy. So we tested this just to test the glutamine hypothesis. And I said we put the animals into therapeutic ketosis to target the glucose. And then on the very bottom you see DON. It blocks glutaminolysis, which is the pathway by which the cell can get energy from glutamine. So you take away glucose and glutamine, and now you make the cell vulnerable because its antioxidant system is going to be corrupted. And then you go after it like that. So here's our experimental design. And you know, this experimental design that we built in the animals, we're now using them in human clinics. So when clinicians treat patients with cancer, the first thing you should say if you have cancer, can I see your protocol for treatment? Well, you want to see I want to see what you're going to do, why you're doing what you're doing, and all these other things. So we do this, and I'll show you from the human how we set it up in human from going from the mouse to the human. So in 15 days, the animal with the high carbohydrate diet, they're dead. They're not dead, but we have to terminate them because for the Animal Rights Act and all that stuff. You don't want them to suffer. So we stopped the experiment at 15 days because the guys eating the high carbohydrate diets, their heads are swelling and you can't walk. The guy's so we put him on therapeutic ketosis and then pulse, 6, 8, 10, 12, pulse with the glutamine inhibitor, and then we compare the brains in the two groups of animals. Nice that we've engineered these cells to give off light when they're alive, and when they're dead, they give off no light. So on the top, the high-carb diet guys, you see the brilliant light, high light. That means you got a lot of living cells. In the diet group, yeah, there's living cells. We weren't able to kill those cells with the ketogenic diet, therapeutic ketosis. Stop their invasion, but we didn't kill them. Didn't kill them. Just remember that. When somebody says to you, Seyfried says you can cure cancer with ketogenic diets, here's the data that says I didn't. Okay, so we have it now at the bottom when we put the therapeutic ketosis in with the drug, no light. Okay, so what was that all about? And then we found remarkably that the delivery of the drug to the tumor is 2 to 3 times better when they're in therapeutic ketosis than when they're not in therapeutic ketosis. So one of the big problems in brain cancer is getting through the blood-brain barrier. We think we found a way to do this. So now let's look at the tissue, because ultimately you want to look at the tissue. Were you successful? So here's the high-carb diets, and you see the dark blue on the left and a little bit of light on the SDUR group. Those are the thousands and millions of tumor cells that are invading into the normal white part of the brain. And they're packed on top. They can't grow any faster. It's a mob scene. They're just piled and piled on top of each other. The metabolic theory, the KDR by itself, you can see how whitish the white, that means they're not invading as much, and they're not as densely packed. But we're not slaughtering them. On the right, KDR plus DON, that's all dead cells. Those are cells that were slaughtered. They're all dead. And that confirms why there's no light. Light means living, okay? No light means death. And are you sure they're dead? Yeah, they're dead. And then if you look at the survival of the animals that got the combined treatment, the drug with the glutamine inhibitor, lived. And we're still working to push it out further. We want these mice to live 100 days. They're out about 50 now. We're still working on dosage, timing, and scheduling, but they're living far longer. They're much healthier. And here's a different kind of a brain cancer. Another one, and you can see the blood vessels in the left side. The ketogenic diet, therapeutic ketosis, removes the blood vessels, anti-angiogenic, and then you have mitotic catastrophe. That means the combination of the drug and the diet kills a completely different kind of brain tumor, and we think they're going to kill every kind of a tumor using this strategy. And then because they can't eat, there's nothing left for them to eat. You pull the plug on their glutamine, you pull the plug on their glucose, and you imploded them, and the rest of the cells are healthy because they're burning ketones. So we built this concept called Press Pulse Novel Therapeutic Strategy for Metabolic Management of Cancer. The authors on this paper are Dr. Yu, he's an oncologist from George Washington University, Joe Maroon, team surgeon, neurosurgeon, team surgeon for the Pittsburgh football Steelers, and Dom D'Agostino, who works on hyperbaric oxygen and also metabolic therapies. So we want cancer patients to come into the clinic, put them into therapeutic ketosis. You see the guy, the Vitruvian man symbol, he's red. He's got metastatic cancer. He's in bad shape. Then you bring him into therapeutic ketosis. Stress management, extremely important, just like we heard from Pam last night. Got to have stress. Stress elevates blood sugar. You reduce stress, blood sugar goes down, and these therapies work much, much better. And there's a lot of ways to reduce stress. And then we pulse, bing, bing. We use various pulses. We hit with low-dose glutamine, glucose inhibitors, and then we use hyperbaric oxygen as a replacement for radiation. Hyperbaric oxygen, we think, will kill cancer cells the same way radiation does without causing harm. However, if the patient is not in therapeutic ketosis, hyperbaric oxygen will not work. Hear that? Okay? The patient has to be metabolically prepared before using hyperbaric oxygen. So it's, again, dosage, timing, and scheduling. We can kill cancer cells non-toxically if we understand the biology and the systems that are driving the beast. Now, this is an example of the tests we did on a patient in Egypt, because we can't do these studies in the United States presently, because they're against the standard of care. So here we took this gentleman. He was a 28-year-old farmer, corn farmer, and he came in, his whole left side was paralyzed. He was a metabolic mess. He had triglyceridemia, he had all kinds of other issues. So we put him on a 3-day water-only fast, and then 21 days of a very restricted ketogenic diet, and then we debulked the tumor, my colleagues, not me, await cradiotomy, so 24 days after being diagnosed, we had a blood work before we did anything to this guy, took out the tumor while he was awake, transitioned him again, postponed radiation and chemo for 3 months, people yelling, no, you got to do it up front. We were able to say no. Then finally they demanded that we do radiation and chemo, demanded it. You can't get away from it. It's a violation of world law. It's written in granite. So we said, oh, okay, well, how does he do? He did pretty good. He was doing well. He did very well during the radiation and the chemo. And then he went back. He worked in the cornfield. He was back. And then we wrote this one. It's 24 months. It's 2 years after diagnosis, which is pretty good. The guy died at 30 months. Came back in. He had a headache and things, so they did a debulking surgery to find out, and it was all, according to the pathologist, it was radiation necrotic damage that killed this guy. So, but we got him to live 30 months, and look, we brought his blood sugars down. He had a beautiful GKI, and if you see the red little line there on the bottom, you can see the major shift to the right because his tumor was on this left side of his brain on this image. But at the very end, 20 months, it's straight. The guy's doing good. He's back working. And we lost him. What are you going to do? I said to them, what if we didn't irradiate this guy? How long do you think would he would? Would he be like Pablo? Who knows? But I think we should try it. So people say, well, I got stage 4 cancer. They call it terminal. I'm thinking maybe these cancers should not be called terminal cancers. I think we have a blueprint and a strategy by which we can stop this growth and we can manage these people. I don't know if we're going to cure. Who knows? You got to wait, 30? If you die at 90 years old from a heart attack and you had cancer when you were 30, you probably were cured. All you know is what is quality of life? The bottom line, how much longer can I live with a good quality of life? My hair going to fall out? Am I going to bleed from all orifices of my body? My teeth and all these problems that I have. Pablo is still going. He has a very high quality of life right now. I don't know how long he's going to live, but he certainly lived longer than Brittany, and he had a much higher quality of life. He got to see his child born, he got married. Who knows? I don't know. I can't predict the future. All I can say is we have a blueprint to do this. So when we conclude, we say cancer is a metabolic disease. It's not a genetic disease. I dedicated my book to the tens of millions of people that have suffered and died from the toxic treatments of cancer, which includes everybody who suffered like that. I think this is the greatest tragedy in the history of medicine. I think this is the misunderstanding of cancer is responsible for the crisis. As I said, I think we can drop the disease by 50% in 10 years if people knew how to do all this stuff. And we talked last night about all the issues that prevent us from doing this. Cancer cells are dependent on substrate phosphorylation. They ferment. If you pull away fermentable fuels, they can't survive. You don't harm the rest of the body. The press pulse metabolic therapy, which we tried on the Egyptian fellow, and Pablo is doing it, and others, it's non-toxic, cost-effective, and it possibly could resolve a lot of the cancers or make them better. So who do we use? We use people in the United States. We work with guys in Turkey. We have a clinic there that's testing all this out. My colleagues in Germany, Venezuela, where they do electron microscope, Hungary, Greece, France, Egypt, India. I went to China. They're very excited about applying this with their traditional Chinese medicine. We have a group that's now starting in Brazil, in Rio de Janeiro. And more physicians are getting together on the planet and comparing and contrasting their data on patients from different places. How is your patient doing? Yeah, he's doing well. Why don't you try this, try that? And these people are living longer. And if I had time, I'd show you all of these remarkable, I don't know how long these people are going to live, but they're doing better than they would have done from what we can see. So, you know, it's very hard to get money to do this kind of research. You think, wow, you should be getting a lot of money to do this. Wrong. Get a lot of money to study the disease. You don't get much if you want to try to resolve it. I get support from the Foundation of Metabolic Therapies, Joe Mercola, the George U. Foundation. Greg Glassman, owner and president of CrossFit, has become very excited about the stuff we're doing. He's been supporting us. Joe Maroon from Pittsburgh. Ellen Davis, who writes books on ways to do this, my own university. And in the past, we did have money from the NIH. So thank you very much for your attention. Thank you. Thank you. Thank you very much. I appreciate that. Yeah, we have. Do we have time for questions?
Getting the mics right now. Just give it a minute. Here they come.
Okay.
Giselle, over here.
We have some.
That was fantastic. I read the book. The book's fantastic. Get the book. Get the book. Read it slow. Read it slow. It's a little. But it gives you the truth. My question is, when you bring these people without the surgery and everything, when you bring the people into where you got them stabilized, you keep them on the ketosis, correct? You keep them on the ketosis. All right. So for those people who you're seeing people that already have cancer, okay? I think one of the keys is that if you get them before the cancer and you keep them on a ketosis level before the cancer, they're not going to get the cancer. And this is what we do. But we have you're eating out of a can, and that's what you eat. So we have prevention is the key to this whole thing. If you keep them, stop the sugar, stop it. Where we can do this because we're controlling the animal. All right. So you're not starving them. You're just not giving them sugar. You're taking it away. You're giving a zero net carb because you got the fiber in there. So you're giving a zero net carb food, and I think that's what we're working to prevent it.
Yeah, well, prevention is the key.
The prevention is the key, all right? So all pet foods, to make the animals eat it, have natural flavor. Natural flavor is sugar, monosodium glutamate, and aspartame. All right, so number one, a lot of these people that you're seeing have been eating and drinking aspartame for a long time. So when you get these people into your control state, I'm using that instead of remission and all these other words, right? If you get them in the control state and you keep them there, the lower the sugar, the more ketotic they are. They're not really ketotic, they're just borderline ketotic. The lower they are, the lower the sugar, the better your results are getting. Is that correct?
Yes, that's completely correct. The problem is, it's prevention, right? So prevention, people are all risk takers. Some people smoke. Right? So the problem is I have a whole section. If you keep your mitochondria healthy, it'll do just what you said. But people we're in a toxic environment in our foods that we eat, the environments that we live. It's very hard but if you do, you're right. If you get low sugar and elevated ketones, it's very preventative.
And if you're on an organic diet, which is what we do, we put them on an organic diet. We totally control their diet.
Yeah, I agree with you.
We have no sugar,
yeah,
net carb.
Well, don't forget, in primitive populations on our planet, Eskimo, Inuits,
yeah,
and Albert Schweitzer in Africa,
right,
those people never had cancer. They weren't eating, so they had no word for it. So clearly, it's prevention. Yeah, we need glutamine for the body. It's when you have cancer, you got to target it. Prevention is a different issue. You have two things here. You got prevention and you got management. Yeah. Obviously, if you have the cancer, you didn't do enough to prevent it. And then you deal with the management issue, which is responsible for the crisis. How do you lower your GKI, glucose ketone index? Don't eat. Right? Try it. Stop eating for a week. You'll get your beautiful ratio. And then you'll realize what I'm talking about.
No.
The what? The meter is Keto Mojo or Precision X. There's only two companies that make these meters. You get them on Amazon. They're about $35. Precision Extra and Keto Mojo. Precision Extra. Get them on Amazon. I buy them on Amazon myself. Keto Mojo. Keto Mojo. Yeah. Yes.
Hi. I would love to see you on a panel with Dr. McDougall and Dr. T. Colin Campbell. And T. Colin Campbell was here the last time. In terms of animal protein, the animal protein itself, the fat, the hormones, and hormones is what makes things grow. I haven't heard you mention anything about it. It's very glucose focused.
No, glutamine.
Glutamine, yeah, and I watched the panel last night. I see glucose is the big thing, the sugar. And I looked at Dr. McDougall's Does Sugar Cause Cancer video, and he says no. And when we talk about, so I want to know how you feel about the animal protein, and T. Colin Campbell has shown, and you can look at statistics that have shown the countries with the highest animal protein consumption with the highest rates of all different kinds of cancers. The World Health Organization has labeled cold cuts bacon a Class A carcinogen. I think there's a lot of focus here on sugar, and I'm not sure if it should be there. And I just want to say, there's a couple other things. That when we're looking at Warburg's, the mitochondria not getting the oxygen and the respiration system, McDougall attributes it to the fat clogging it. That every time you eat a fatty meal, your oxygen level in your cells goes down by 20%. So what about animal fat, animal protein, animal hormones? And when you say high carb, what exactly do you mean? Do you mean refined high carbs or high carbs, whole food? And if you're putting someone on a keto diet, I automatically think of animal protein. What does it mean when you put a patient on a keto diet? What are they eating daily?
Well, you've got several things overlapping here. Okay, you've got several convoluted things here. All of your points are exactly correct. Proteins, the ratios of proteins, fats, and carbs in a particular diet is very important. But we get through all that by looking at the ratio. Look at the ratio. As a matter of fact, I know Colin Kemp, we had a long discussion about this. And he's right. When we gave our mice too much protein, the tumors started growing much faster. When we cut the protein down, the tumors started growing slower, exactly supporting what he said. So it has to do with the total calories coming from the macronutrients. A long time ago, so it's everything is the calorie content and the kinds of calories you're eating. So people, if you want to know the answer to your question, do all these things that you said, and every time you made the change to this, to that, what did it do to your ratio? And when you come to the realization that you are now in therapeutic ketosis, you know what you can and cannot eat. So therefore, it gets through all that bushes and all the convoluted things. What is the ratio? Because when the body is in therapeutic ketosis, the tumor cells can't grow fast. We have shown that because we are capable of doing it in an animal that has no ability to choose things for himself. We have everything regulated. Humans are different. They cheat on themselves. Can you believe this? I can't answer the question. You're different from me. The diet that you eat may deliver a very different ratio than the same diet we eat may deliver a different ratio. So that's why the ratio is important to know. And therefore you get through all this other stuff. People say, I can't eat that. Is it too much protein, too much? What's your ratio? I never measured it. How the hell do you know anything? If you don't know the ratio, then you can't answer the questions. Then once you're in therapeutic ketosis, then you can add more protein if you like. Vegan, do this, do that, and see what it does to your ratio. And then we did this on wine. People get into therapeutic ketosis. Can I drink? I mean, a lot of people in the society like a little nipper every now and then, right? Me included. That's why I did the experiments. So when I get cancer, I know what I can and cannot drink. You got to know, what did it do to the ratio? Dark red wine, nothing. White wine, bang, no white wine. Beer, bang, no beer. Bourbon, oh, bourbon, loaded with sugar. Scotch? So you got to know, and I said it comes right back. Your questions are covering a lot of extremely important issues that confuse so many people. And we try to get through all that, the biochemistry. But your points, this is one of the great confusions about ketogenic diet. What you have just articulated is actually the core of one of the stumbling blocks of moving through the field. What we're trying to do is simplify it so we can address, and then we can pick off for each individual what those are. Sorry.
Mr. Seyfried,
Always in the wrong
may I
spot.
ask a question, please?
Yeah,
Thank you so
in the
much.
back there.
Yes, for your research and all the hard work that you do. Thank you. I had breast cancer, and your treatment is one of the treatments that I use. Naturally, I am completely cancer-free. But thank you. My question is I've been told that I have MTHFR mutation, and I don't know how that affects this whole Krebs cycle and how to maximize my energy. Can you enlighten me on that?
No, I don't know anything about the mutations. We have discounted them. They're irrelevant. They don't mean anything. Okay, everything is energy. As I said, in our normal cells, we have the same mutations that you mentioned in our normal cells that aren't cancer. So what does it mean? If your tumor has that mutation in the breast and your colon has the same mutation and has no effect on the colon, what does it mean? So I tell you the mutations are you know what a red herring is? A red herring is something that diverts your attention away from everything else. So you get 1,000 silver herrings and there's one red one. Everybody looks at the red one. The gene theory is like that. Everybody's looking for the gene, and the genes are really irrelevant. Yeah. I'll get this gentleman
Oh,
here.
I have the mic, sorry. I want to thank you. I want to thank you very much for your speech, your talk. Not only was it informative, it was enlightening. I know a lot. I'm a healthcare practitioner. I've been studying cancer. I've been with cancer in Tijuana, Mexico. And the glutathione, everything you've said is 100, and the sugar is 100% accurate. I was told 20 years ago, you put sugar, I was shown by a doctor, with the cells. I was in his lab. It showed how they grow within seconds when the sugar attaches to cancer cells. Now, I am hearing some people that are saying things contrary to you. They are not professional in the sense of maybe they've heard other talks about Campbell, and I support all types of diets, but we're here today to thank you because you've taught us today, first of all, how our wonderful establishment doesn't allow certain testing here in the U.S., so you have to go out of the U.S. I am not going to say anything negative about our system because we all know how negative it is. And this man is brilliant. He's fighting for us as citizens against the medical establishment. So I don't like the fact that people are challenging. Yeah, there are a lot of things to be saying about an animal protein, but you are discussing and giving us a new concept with this metabolic theory. You're showing us wonderful things. And I think that if what you're speaking to layman people here, and what you're saying is very brilliant. So I think people have to have an open mind and understand different concepts because it's not all about this. You have a brain, you know you're not going to have protein with hormones. So I don't like anyone challenging this doctor who I've never met and never heard of before. I'm very happy to hear this. I will be getting your book because we have to look at facts like you gave, and we have to look at solutions, and we have to take care of our health because that's all we have. And I really am blessed that I came here to hear this, and I thank you for sharing. And let's all keep an open mind and not be stuck in our theories of what we know, because this man is brilliant. Thank you.
Thank you.
Okay.
I want to point out one. I know you have a lot of questions. In my mind, there's one way, a way, that we can change the system. Are you ready to listen? Okay. We have fragmented the cancer into several different diseases. If you go and you say cancer is many diseases, so therefore that justifies a breast cancer foundation, it justifies a lung cancer foundation, it justifies a brain cancer foundation, it justifies the existence of separate foundations. As long as the foundations are separated, it's unlikely that we're going to move the field forward. You need a united foundations of cancer. Just like you have the United States of America, you need a united foundations of cancer. Recognizing that everybody's cancer is essentially the same, it just happens to strike different tissues. Once the foundations come together, they will do the same thing that happened with the AIDS crisis. You understand what I'm saying? It has to be a unification of the cancer foundations. You don't need to know all the sophisticated biochemistry like we do. You just have to know how do you solve the problem. And one of the ways to solve the problem is unite the foundations. When they get together, they'll put more pressure on Washington than the Million Man March and all these other things that you see going down there. We stopped the Vietnam War from the people going to Washington. As long as we keep the foundations separated, we'll not be able to break the barriers. It's just that simple.
Okay. I have a question. I have a comment. My name is Yolande Smith, and I'm one of the pioneers of the health movement since the natural hygiene. I don't know if you know about Natural Hygiene Society.
I don't know much about that.
Okay. So it's where Dr. Furman came from, Dr. Goldhammer, Dr. Colin T. Campbell. This is from Dr. Herbert Shelton. He was like the father of the movement. Anyway, I've been involved with the health movement for 32 years with passion and 4 hours every day. Me and another woman here named Wendy Harris, we've been involved with that movement and learning 4 hours myself, 4 hours every day. I listen to all kind of health conferences, lectures, all over the world. I my first eye-opener was the documentary called Diet for New America by John Warburg and Dr. Clapper. And since then, my journey started with Dr. Furman. I did a conference with him in New York a few years ago, and Howard Lyman and all these people. So that led me to True North Health, which Dr. Goldhammer, who is here today, is the director. Dr. Alan Goldhammer is going to be speaking tonight at 7. I've been to this clinic 3 times, and I sent 30-something people to this clinic. I sent people to Hippocrates Health Institute, about 5 people. They all recover from terminal cancer. There's a woman who came to me, Sloan Kettering gave her a death sentence of 3 months, and she was crying. And I sent her to Hippocrates. Her name is Radera Lloyd Bay. And she went to Hippocrates 3 days later, and it's been 10 years.
Yeah.
And she's alive. But
Well, I mean,
yeah,
you're gonna have a lot
my
of
thing
issues
is,
like this.
you spoke about the problem, all the technical aspect of cancer, but I didn't hear much about the solution. And I believe the human body can heal itself if we give it fuel. That's what I learned over the 32 years. I can have cancer right now, go to my house, put living food in my kitchen, and after 2 to 3 weeks, that cancer will most likely go away. Okay?
Yeah, well,
so,
maybe.
True North Health, Cornell University did a study that shows that 98% of people reverse their disease from fasting with major disease. My question to you is, you spoke about the keto diet, which is like a meat. It's not, by the way, that lady that said we're criticizing you. People must have a critical thinking. You must be a critical thinker in order to find answers, solutions. So you spoke about the keto diet, and I think that keto diet can damage people's kidneys and liver because it's high in protein and fat, and there's no fiber in animal protein.
Okay, let me answer your question. The ketogenic diet, she is right. If you eat too much of a ketogenic diet, you're going to get insulin insensitivity and you're going to damage. If you eat a dollop of it, a dollop, you know what a dollop is? A little bit. Huge difference. You understand? I agree. If I sat down and ate a pound of butter and a bucket of lard, I'm not going to be in good shape. If I lick the lard spoon and sniff the butter, it will be a lot better. You understand? It's one of quantity. Yeah, we did it. We've done all those studies in the lab. Yes, you got to lose a lot of weight. You got to measure your glucose. We did all that. I don't know about that.
My question is, you talked about you talked about the different stages of your of the people on your treatment, and when you put them in ketosis, they didn't have the cells regenerating and
yeah, they didn't grow as
reproducing.
fast.
But then you talked about you inhibited the glutamine with the drug. Is there any way for a person who doesn't go to your center to be able to
I don't have a center.
limit
It's a university.
to someone who is not receiving a drug? Is there any way to inhibit glutamine from a layperson doing it at home like you can do with glucose?
Well, I don't know about that. Those drugs, EGCG, it's a green tea extract. It doesn't work as well. You know, maybe the neoplastons from Dr. Brzezinski's, those things might also inhibit some glutamine, but I haven't tested it yet. Once people are, and believe me, the pharmaceutical companies are very, very aware of the glutamine issue. Very, very aware, and they're trying to patent drugs that will do that. But none of them seem to work as well as Don. And Don was treated for cancer, but didn't work so well because they weren't targeting the glucose. So these drugs are coming, and they will be here, but you got to make sure they don't harm your body. Okay? So unfortunately, there's no clinic in the United States that you can go to right now that will do what I just said. But I just wanted to let you know it can be done. If they did it, we're going to get the results. That's my prediction. Okay.
Hi.
I think, do we
sorry.
have time for more questions? Or I'll be happy to.
I have a quick question about, I seen on your slide there, water fasting. Is there any particular water you recommend? And do you know anything about dry fasting, and would that work? Or is that
You mean
dangerous?
dry, with no water?
Yes.
Just
you
no,
know,
no food and no water?
no food, no water for 3 to 5 days.
That study was done in Italy in the 1800s. The condemned prisoners. Okay? The longest a human being can live without food and water is about 17 days. Okay? They die from dehydration. But when you have water, you can live a long time. And those studies were done in May's prison camp in Northern Ireland. Bobby Sands in that group, okay? They have all the records on how long it took these young, skinny Irish guys to die from starvation. Starvation is a brutal way to go, believe me. So, yet my friend George Cahill, when he was alive, he fasted a 400-pound guy from the mail department, lost his job delivering letters, 6 months on a water-only diet. He lost like 280 pounds. But you know, you don't have to supplement with vitamins and minerals until after about 20 days, because the bones contain all the minerals, the liver contains a lot of key vitamins, and there's a lot of fat-soluble vitamins. So when you start fasting, the vitamins come out of your fat. So you really don't need to supplement until after about 20 days. Then you might want to consider vitamin C and these kinds of things.